Morning, everybody. Thanks, Laura, for the invitation. It's really exciting to be here and to share the work that we're doing in the context of ovarian aging and ovarian rejuvenation. So I'm Vittorio. I'm a faculty at Stanford, and also, as a disclaimer, I'm also co-founder and head of research of Turn, and I also very recently joined the Reverse Aging Advisory Board at the Beauty, which is trying to do some yeah. Okay, so, well, at the high level, what we're trying to do in the lab is, on the one hand side, to model and understand human embryonic developments and gametogenesis, with the goal to not only to understand it, to model it, but also to apply it in a context that is different from embryonic development and gametogenesis, which is tissue rejuvenation and improving health span and a healthy longevity, in particular in women. So my lab, so why are these two things connected? Why reproduction and aging are connected? I think they are connected in many different ways, and so I will try to explain why. But the first and most important reason, which kind of struck me as a sort of epiphany when I started thinking about actually the problem of aging. I come from developmental biology and reproductive biology, so that's my training. But when I started thinking about the problem of aging, I really realized that nature had already figured out a way to not age. Okay, so we do age as single individuals. We definitely see that happening every day, in the mirror. But if you think about that, species do not age. Individuals in the species age, but the species doesn't age as a whole. Every time a species, goes through reproduction, the new generation is born at age 0. So there is no vertical transmission of whatever age has occurred in the parents, and this has gone on, for billions of years, and actually life hasn't aged for 3.6 plus billion years. So, what am I trying to say here? I'm trying to say that in reproduction are the secrets and the principles actually for not aging. The problem though is that happens in embryos and in germ cells. So what we have to do is to learn those principles, to understand them better, and apply them to non-embryonic and non-germ cells. So that means the soma, the body. So that's the first link, which I think is the most compelling one. And then I will get to the second in a minute. Leveraging those, so by learning basically these principles, so where's the pointer? It's the top one. My lab has discovered or has pioneered a process which we called epigenetic reprogramming of aging, which in a way is inspired by the zygotic reprogramming and the germ cell reprogramming, so the epigenetic reprogramming that happens in embryos and in germ cells. So we call it ERA, epigenetic reprogramming of aging, and it's in a way it's one of the first examples of how you can repurpose epigenetic reprogramming in the context of embryonic development and germ cell development and apply it to somatic cells. And so, to make a long story short, what we have shown, and this is a work that was published in 2020, we have shown that you can take a dysfunctional cell, an old cell, and deliver inside the cell a cocktail of mRNAs that encode for reprogramming factors that, if expressed for a very short time and in the optimal dosage, can make that old dysfunctional cell into a youthful functional cell, keeping the identity unaltered. It's very important because we're not making iPSCs, okay? iPSCs are great for other reasons, but we're not making iPSCs. We're making, for example, an old skin cell into a young skin cell, okay? And we have shown that since we're working at the epigenetic level, we are actually tackling or impacting most, if not all, of the hallmarks of aging. So now the cells are not just youthful because they are epigenetically youthful or because they are molecularly youthful, but they're youthful because they're functionally, physiologically, and also molecularly youthful. Okay, so, and I'm actually proud to say that my lab has been really one of the pioneers in this field. We have been the first to show this in human cells. And now, as you can see, the field has really exploded, and there has been multiple examples of how this concept of epigenetic reprogramming or partial reprogramming, transient reprogramming, interrupted reprogramming, there's different names for it, is really a reality, okay? And it works in mice, in rats, in humans, in non-human primates, in muscle, in a variety of different tissues. So we just published a review on the topic, so if you're interested, I mean, there's a lot of information there that you will find. Okay. Now, the second reason why aging and reproduction are intimately linked, and I think we have already heard a beautiful introduction on the concept, but is in particular in women, is the fact that the aging of the ovary, which is a reproductive organ, impacts the aging systemically of the body. And so I'm going to go through this very quickly because, again, you got an excellent introduction before me, but we know that the timing of menopause is strictly linked to the susceptibility to age-related morbidity and mortality. And I'm talking in particular, for example, in cases like POI and POF, where women undergo menopause in their 20s, late 20s, or early 30s, right? So we're not talking about physiological menopause in this context. But it's also true in physiological menopause. So there is a small study suggesting that for every 1 year of increase in menopause age, there is an age-adjusted mortality rate that is decreased by 2%. Or, together with all we have heard before. Now, if you use animal models, and there is also very compelling evidence suggesting this. So, if you take mice and you do ovariectomy, which is in a way to mimic menopause, even though it's not really a menopause model, but there is so basically you remove the ovaries, and this leads to shortening of lifespan, reduced health span, and it's also this process is also affecting a variety of different organs and systems: the bone, the muscle, the brain. But strikingly, if you take now an old animal, you remove the ovaries and you put in young ovaries, this has an impact on lifespan and health span across the board, okay? And what's even more interesting is that if you do the same procedure, but if you remove the germ cells first, so you kind of ablate the germ cells from the ovary and you transplant this young ovary, which is germ cells depleted, that effect is even enhanced, suggesting that there is an interesting, dual function of the germ cells and the somatic cells that needs to be investigated. So why is this exciting? Well, it's exciting because I think, as we know, the ovary now, as it ages, impacts two important things in women. Why? Because the ovary is a very special organ. It's the organ where the oocytes and the follicles mature for the following generation, but it's also the organ that communicates with the rest of the body and tells the body very important things. So, for example, it communicates, well, be ready because an oocyte got fertilized, so you need to be ready for gestation and for implantation. But it's also systemically balancing the well-being of the woman through hormonal control and other things. And so, we know then that with ovarian aging, two things happen. Well, on the one side, there is a fertility decline that, again, we have heard. I will go briefly through this. So there is a decline in follicle numbers, follicle quality, and so on and so forth. But there is also a systemic process of aging that is impacted by the aging of the ovary. And this now has been shown by a variety of different groups in different animal models. I mean, there's plenty of evidence now that is really telling us, well, it's about time to do something about it, right? Okay, so the central hypothesis of my work is that the ovaries play a key role in enhancing or delaying the process of aging systemically. And so the question and the mission that we have is, well, on the one hand, can we prevent or even reverse ovarian aging? And by doing so, we could potentially impact women's health span, postmenopausal health span. But also on the flip side, because of the dual function of the ovary, by rejuvenating the ovary, can we also prevent or reverse the loss of follicles and the quality of follicles. And so the way we are doing this is two ways. So the first one is by generating ex novo new ovarian tissues from stem cells using iPSCs, for example. So this is a tissue engineering approach to generate novel ovarian tissues from immuno-matched stem cells, which obviously has implications also for in vitro gametogenesis, of course. But the second approach is also taking what's there, what's existing there, and just trying to make it younger using the ARA technology. So these two things are kind of have the same goal, but they are in a way complementary strategies. And so I'm going to share some data very quickly. So regarding the first part is published data. It's a paper that we published a few months ago, Initial Communications, which is aimed at the in vitro gametogenesis in a way, or the formation of new ovarian tissues from stem cells. So in this paper, it took us 8 years to get here. But basically what we did was to develop a 2D, a monolayer platform to generate practically unlimited number of primordial germ cells from human pluripotent stem cells. So in about 3.5 days we can generate between hundreds of thousands to millions of PGCs. Just very quickly, from a transcriptional standpoint, the PGCs that you see here, so they're called the PGC-LCs because they're primordial germ cell-like cells. The reviewers were very adamant about that. So we did a single-cell RNA-seq analysis, and we confirmed that the PGC-LCs that we generate in vitro are transcriptionally similar to the fetal germ cells of the class 1 that were characterized in a study from a Chinese group. So basically what they did was to take human embryos, sort out PGCs from week 5 to week 23 approximately, and they classified based on the gene expression the FGCs in 4 classes. The 1, which is the mitotically active ones, the 2, which are the ones that are committed to germ cell fate, and then the 3 and 4, the ones that are more advanced and meiotically competent. So our PGC-LCs are very similar to the early PGCs, the mitotically active ones, the ones that form in the embryo and then colonize the genital ridge a few weeks after. And this is just fancy ways to prove that. So you see that the Pearson correlation analysis is the highest, comparing the PGC-LCs with the FGC1. And if you also do a t-SNP plot analysis, which is again another bioinformatic tool, you see that the PGC-LCs in vitro, which are the green ones, are very similar or very close to the FGC1s, and there is a subpopulation which is practically overlapping, suggesting again that there is transcriptional similarity. So where we are with that? Well, I think we have already very successfully recapitulated this part of the formation of PGCs. So now what's left, quote unquote, is really to try to commit the PGCs into a germ-like fate, which potentially, if we get here, this could already be on its own a potential clinical product, because we were discussing yesterday with Josh, if we could get the cells to an early meiotic competent state, maybe that would be enough for them to be transplanted into an aged ovary, and potentially, instruct the ovary and tell the ovary that there is more primordial follicles around, and this could potentially have an impact on its own. Okay, and on the second part, so which is the rejuvenation of the ovary, so I'm sharing some unpublished data. Okay, so brand new data that came just a few weeks ago. So again, remember that what we're trying here is to try to rejuvenate the existing ovarian tissues by the expression of those factors that are implicated in the rejuvenation, so the so-called Yamanaka cocktail or versions of that. So of course this is done in animals, and so we're taking advantage of a transgenic animal where by just giving doxycycline, which is an antibiotic, we can induce the expression of 4 of the 6 factors that we have described before. So you see that up here in the absence of dox, the genes are not expressed, but in the presence of dogs, the genes are turned on. Okay? And so, what did we see? Well, early but very exciting data. We have seen that after a month of expression of those factors, there seems to be, in the treated ovaries, there seems to be an increased number of primordial follicles, secondary and antral follicles. We do not know yet if this is due to a delay in the maturation process, so if the oocytes somehow are stuck, if there is a proliferation of primordial follicles that then results in a higher number of secondary and antral follicles, or what's going on. So early evidence, but very, very exciting and very promising. But another piece of evidence, which also could be interesting, so we tested in parallel the effect of VCD, which is in a way, it's killing, it's a molecule that is used in the literature to kill specifically the primordial follicles, so the very, very early follicles in the ovary. And you can see here, just in the controls, you see that if you inject the oil, you have a residual number of primordial follicles there that are stained with these two markers. But in the presence of VCD, after just a couple of weeks, the follicles are all gone because they have been killed specifically. And of course, what you are left with is only the secondary ones that got engaged in the maturation before the VCD was given to the animals. And the quantification is down there. But what we are seeing is that when we give VCD and the 4 factors at the same time, there seems to be a preservation of the follicles. So the primordial follicles don't seem to be dying, as in the case of the VCD alone, suggesting that there is potentially a protective effect of the expression of these factors against the VCD-induced killing. Why is this exciting? Because if this is true, and obviously we need to do follow-up studies, this could potentially mean that partial reprogramming could also impact loss of follicles driven by pollution or by other agents, phthalates and other things that we know exist in the diet. Okay, with that, I want to thank the people in my lab, lots of women, very proud of that. My institution, the Stem Cell Institute in Stanford, the funding and all the people in the lab, and I'm happy to take your questions. Yeah.
I guess I'll wait for the mic, even though I'm pretty loud. A question about the new data that you have. So, are you planning to try to isolate the oocytes from these regenerated ovaries? And how will you assess the oocyte quality between your controls and the ones that you regenerated? I'm asking about your next experiments.
Yeah. We are planning to do a bunch of different things. Well, obviously, we want to characterize at the molecular level. So first of all, we need to also start working with much older ovaries. So these were ovaries that were taken at kind of middle-aged ovary in terms of mouse life. That's 12 months roughly. So we now need to do the studies with the much older ovaries, even though at 10 months the reproductive capacity of the mice, even though they're not going through menopause, but the reproductive ability of the mice is significantly declined. So yes, we want to characterize molecularly, for example with RNA-seq, but also functionally. So do they go through the same, so do they have the same percentage of insemination rate, for example, pre-implantation development capacity, post-implantation development capacity. And the good thing is that we can also do some longitudinal studies on the following generation to see if anything is impacted, in the newborns. Absolutely, yeah, we're planning to do those studies, but we haven't started yet.
Specifically, I was going to ask if you were going to be looking at aneuploidy.
No, because mice unfortunately are a horrible model for aneuploidy. They do not go through aneuploidy that easily as in the human case. So I don't know if that's going to be a good assay for mouse developments, but yeah, we will see.
I'll show you my postdoc's paper.
Okay.
I'll ask you again after that.
Okay.
great talk, thank you.
Thanks.
So I have a couple of questions.
Sure.
So for the PGCs,
Yeah.
I noticed there, in your protocol you had basically Wnt inhibitors. And I was kind of curious, thinking translational for therapeutic, obviously Wnt inhibition reduces bone mineral density, so it has a lot of metabolic effects.
Yeah.
Are you thinking this is still research, and for translational it will have to be a local delivery, or what's your thinking around that kind of for a systemic effect?
Oh, sorry. I should clarify that that's in vitro derivation of germ cells. So we do everything in vitro, and then once the germ cells have been made, that potentially you would transplant those into the ovary.
Okay, so it's kind of local.
So there is no in vivo stimulation of this. So yeah, it's like a cell therapy type of approach.
Yeah, so I was just kind of curious if you envisage it as like someone will take a pill, or you envisage it as like in vitro. Okay, that answers.
Yeah.
And then the second question was regarding the rejuvenation of the ovary.
Yeah.
Initially, when you made the point about ablating the ovaries of the germ cells and putting it back, so I was curious if you're aware of any comparisons with HRT therapy. So, what's the effect of HRT therapy in terms of longevity and lifespan? Does that kind of work as a ovary without germ cells? Yeah, what's your thinking on that?
I am not aware of any study, in that domain, so I don't know if anybody in the audience is aware of that. I've never seen anything that targets the germ cells and then, studies the impact of that in terms of longevity. Josh, is anybody?
No, and the comparison with HRT, there tends to be the modification of risk of terminal diseases, right? So where ovaries are working longer, they're offsetting your risk for certain cancers, are the reason for extended longevity not attributable to working ovaries versus hormones that you've lost.
Well, that's why we're here.
That's right.
Yeah, on your transcriptomic data, there's a set of genes that are really differentially expressed between both on the top of it. I really got interested in what are the differences. I mean, it's always great to have like similarities, but that like this set of genes, if you go back, that actually you didn't annotate. Yeah, on top, it's like green and red, and so yeah, exactly.
Yeah. No, at the top of my head now I don't remember what those genes are. It could be that those are mitotically involved genes, because of course these are, keep in mind that there is heterogeneity, of course, and these are coming from different embryos at different stages, from 5 weeks to 23. But they are all classified as FGC1 based on their transcriptional profile, but that, of course they're very heterogeneous. So my hunch is that these genes potentially are implicated in the cell cycle.
Okay.
And of course, in our case, the cell cycle genes are more highly expressed because the cells are in a very active proliferative stage, and these are less so. But I will do my homework and I will go back and check.
That was actually the same question I had, but just like the genes that aren't overlapping if you did gene ontology, but you just answered.
No, I haven't, so yeah, I will.
Okay, just one last one.
Sure.
You have a slide that was wanting to put the germ cells back into the aged ovaries.
Yeah.
So I'm surprised because are you planning to also have some somatic cell components as well? Because we know that the primordial follicle, if you put germ cells there, there's no somatic cells around it. The primordial follicles, the germ cells will just die off.
Good point. Thanks. I forgot to mention that in addition to the PGCs, we're trying to also generate granulosa cells from iPSCs,
right,
with the idea that then to reconstitute an ovary. In reality, what I would like to do is to test both, like going back to the idea that maybe the granulosa cells are the one that are the main driver of this lifespan and lifespan extension, which still needs to be confirmed. But the idea would be to transplant the young granulosa cells on their own, or maybe transplant the follicles in the ovary and see if there is a difference.
It's like so actually we did this work already with my collaborators, and we're just waiting to be accepted somewhere. So it's on BioRix. So what we do is that we actually had a platform. We took out younger granulosa cells, and then we try to grow it together, reconstitute it with the aged oocyte. And what we see is that the younger environment does change, the age of oocyte. I mean, of course, in a mouse. So that's why I was thinking, when we need to put these germ cells back into the aged ovaries, that is quite important to have it. But we have sort of shown that it really can help rejuvenate but yours is germ cells, so it's a different level altogether.
Thanks. No, that's very interesting. Yeah, thank you.
Another question?
So I have time for one?
Yeah.
First of all, absolutely brilliant work, in my opinion. Brilliantly delivered also. Quick question. How can we make this a future therapy? What are the roadblocks you see ahead? Is it just funding? What do you see as challenges? And how much time do you think this can become a reality?
Okay, well, funding for sure. I mean, that's number one, as always. So I tell you very honestly, I think that based on what we know, both in terms of the importance of the ovary in the systemic aging, and I think also the data that my lab and others have shown about partial reprogramming, but also about generation of new ovarian tissues. I think that there is a unique opportunity here that by targeting one organ, this is really unprecedented. People are thinking about taking a pill that maybe reaches every cell in the body, or people have asked me, do you think that the partial reprogramming will ever be, reprogramming every cell? I don't think so. I mean, that's really impossible. Imagining that you can target every cell in the body and then you have to reprogram them in different ways because, of course, every cell is going to be different. But if we understand that maybe there is one organ or two organs that are the major drivers of aging, and guess what? The ovary is one of those. Maybe you have an opportunity just by simply focusing on the ovary to really impact a lot of things. So what I'm trying to say here is that there is a huge potential that I think is within reach. Now, how do we get there? Well, of course, funding is important because, most of these, there's still a lot of things that we do not know. We were discussing yesterday with Josh, why the oocytes age? We know that they become unemployed with time, but why? Is it because of cell intrinsic mechanisms? Is it because the granulosa cells around them age, and then there is a wrong communication? We still don't understand. So there is a lot of work that needs to be done in terms of understanding, and there is also a lot of work that needs to be done into bringing this proof of concept into a clinically translatable option. Because of course, let's not forget that even in the best case scenario that we can just rejuvenate the ovary, how do we do it? Do we deliver the mRNAs inside? Well, that's challenging from a delivery standpoint. Or maybe as we learn what we learn, maybe we understand that maybe there is a small molecule that could be used to achieve the same effect without going through gene therapy or gene delivery. So again, it's a long way to say that I think we could be we could be there fast if the resources are there to support basically the research. So I don't know if I answered your question. You did?
Family first.
Family first.
Thank you so much.
Thank you.